Now, this is a complicated slide. I promise not to belabor it, except that there are two really outstanding scientists, Doug Hanahan and Bob Weinberg, who identified the 10 characteristics that are present in all cancers, irrespective of what cancer it is: lung cancer, colorectal cancer, prostate cancer, pancreas cancer, breast cancer. They all have these characteristics that are regulated by these genes I've been telling you about. And what has been exciting has been the fact that we've now been able to identify these genes with the breakthroughs that have happened in molecular biology and biotechnology. We can dissect a cell at a level that we could never do before. We could take apart the DNA and actually sequence it and see if the sequence is normal or if it's broken, and see if critical genes are broken, and then think about what we might be able to do about that. Now, the traditional cancer treatment approaches still are a mainstay of what we do for cancer. The initial approaches to malignancies are in the hands of our surgical colleagues who try to remove the tumor in its entirety. So you will hear about the surgeons going in, resecting a large area of whatever the organ is where the problem arises, unless the tumor is too big for a resection or it's too close to critical organs. And they'll look for something called clean margins, which means the tumor has not extended to the area that they've cut out. So they'll come back, and one of the unfortunate things that happens in the therapy of cancer and cancer patients is they've been told by their surgeons, and not through any malice, we got it all. And the truth is, you've got everything you could see. But these tumor cells in that tumor mass have the ability to invade the vasculature and the lymphatic system within the tumor bed and get access to other areas of the body simply from that. And so even some small tumors that are wired to behave aggressively and invade like that can be problematic and cause a recurrence. And it's metastatic disease, generally not the primary tumor, that ends up being the clinical problem or the thing that may kill a patient. It's not the primary tumor, with the exception of brain tumors, where it's an enclosed area with vital function. And if that primary tumor continues to grow, it can cause significant problems. But if breast cancer were just restricted to the breast, you would think simply removing the breast would take care of the problem. The difficulty is those breast cancer cells can travel outside the breast to other organs and start to develop and proliferate there, because their proliferation signals are all messed up. Their motility signals, their signals that allow them to move, are all messed up. They're signals that allow them to recognize where they're supposed to be in the body. That, you remember I told you about the embryo and everything going to the right place? Those are all messed up, and any of those can be mutated to cause this problem. And it is the disease that's spread outside the breast that is the challenge that we try to treat. So our surgical colleagues try to remove the bulk of the tumor, which is an important thing to do. Our colleagues in radiation therapy will try to use additional therapy in the initial stages of the disease to make sure the area or the tumor bed is sterile, or any of the areas immediately around it. They can also use radiation therapy in case subsequently something that did escape starts to cause a clinical problem. So they can direct the radiation beam to those areas and cause cell death with the beam. And then the medical oncologist uses systemic therapy. They use what traditionally have been chemotherapies, or poisons, for want of a better word, that kill cells with the idea of killing more bad cells than good cells. So you throw in non-specific cytotoxics with the objective of killing more bad cells than good cells, and that does make an impact. And it does increase cure rates, there's no question about it. But when you think about it, it's a pretty primitive way of approaching the problem. Now, this is just a cross-section of the body, and I only put it up there not because I'm going to give you an anatomy test, but simply to tell you that any tissue in the body can develop a malignancy, from the brain all the way down to the foot. The bones, the muscles, even the cartilage, these all can develop into malignant cells and cause a tumor, and they're called different things. You may hear from patients who've had breast cancer, now I have lung cancer. What they're really telling you most of the time is, I have breast cancer that has moved to the lung, or breast cancer that has moved to the liver. Or a patient with lung cancer says, now I have brain cancer. What they're telling you is, my lung cancer has metastasized to the brain. That is not to say there aren't primary tumors in those other areas, but that's what happens, and any tissue is at risk. The ones that are proliferating more are more at risk. The ones that are proliferating less are less at risk. That's why there's not too many malignant tumors of the heart. And central nervous system tumors are thankfully at a lower incidence than some of the other ones like the gut, the lung, the prostate where cells are turning over, the breast, etc. Here is American Cancer Society data showing the incidence of cancers around. Colorectal cancers are high up there. Cancers of the lung and bronchus are among the highest, and that's because of cigarette smoking and the proliferation of tobacco use in the developing world. So these are global problems, and you can see the incidence of the other cancers. Now, the GI cancers that I just mentioned cover a whole spectrum of organs that are in the gastrointestinal tract, and as I only put this slide in here to make the point that any tissue is at risk to develop a malignancy. This is just the tissues that are in the gastrointestinal tract. You can think about all the other tissues in the body and see the same sort of phenomena going on. Now I'm going to switch gears and finish up my talk talking about a disease that has taught us many paradigms, and it's a disease where I think most of the advances have been made in understanding the basic molecular biology of cancer, and that's breast cancer. Now, in truth, we should have known as physicians a long time ago that we weren't dealing with one disease. But the problem was people said it's breast cancer, it occurred in the breast, it's one disease. Colon cancer, colon, it's one disease. Lung cancer, it's one disease, et cetera, you get the picture. We should have known that it wasn't one disease by virtue of the fact that the patients had very different outcomes. Some patients have the disease, get their primary therapy, and that's it. Nothing else happens. Other patients with the same size lesion, so it's caught early, it's resected completely, appropriate radiation is given, but they have a recurrence and they have a much more aggressive disease. It's possible that perhaps they have a different type of breast cancer that's behaving more aggressively because the different wiring is broken. I hope I've convinced you that there are several roads to Rome to go from a normal cell to a malignant cell. There are multiple pathways that can be broken, and some of them in combination. What we do as medical oncologists is throw in these nonspecific bombs in combination to try and kill those rapidly proliferating cells, but we also affect normal cells, the bone marrow, the gastrointestinal tract, hair loss. What I tell my chemotherapy patients is theirs will come back, mine will not, so that's the only upside of it. But the problem is that you don't want to be that nonspecific if there are better ways. Now, the disease breast cancer, this is statistics from the World Health Organization, it's up to 1.6 million now. These numbers are a bit old. New cases each year develop globally. 1 in 10 cancers that occur will be breast cancers. 1 in 5 to 1 in 6 women will develop this disease. And it's very much related. The first time the United Nations and the World Health Organization has declared a non-infectious disease a medical epidemic is breast cancer. And that's largely related to what I told you. In developing countries, just clean water and access to sanitation has made people live much longer and not die of infectious disease like cholera and other infectious ailments. So they're living into their 5th, 6th, 7th decade, and the incidence is going up. This is the mortality rate. More than 600,000 cases, 600,000 patients globally die each year. Here are the data for the U.S. 200,000 new cases diagnosed and about 39,000 deaths. This is unacceptable. We have to turn this around. In the past, as I said, and as Jim said in his introductory comments, have been dominated by a one-size-fits-all approach. You have lung cancer, you got one of a couple of combinations of chemotherapy. Breast cancer, you got one of a couple combinations. Prostate cancer, you got some types of therapy that were thought to be specific for that. But everybody got pretty much the same regimen. And yet the outcomes were very different. So we should have begun to appreciate that maybe there were different subtypes within each of these diseases. Now, breast cancer sort of led the way, and you'll be surprised that it went back as far as 1899 when a surgeon by the name of Sir George Beecham, he got knighted for this work, said, well, you know, breast cancer occurs in women. Maybe this is a hormone disease or hormonally regulated disease, and the ovary is a major source of hormones in premenopausal women. Maybe removing the ovaries would have an impact. So he had a patient come to his clinic that had advanced breast cancer, metastatic disease, and he did what's called an oophorectomy. He removed both of her ovaries, and there was a spectacular response. All the disease went away. And in those days, you could write up a single case and get it published. And he wrote up that case, and it was published in the literature. And everybody paid attention. But what got very little attention was the third paper he wrote, which is another patient who came to his clinic looking exactly the same way, metastatic breast cancer, young woman. He removed the ovaries, and the tumor just progressed right through everything and didn't respond to anything. Therefore, he was the first to really identify there was a hormonally driven group of breast cancers and another group of breast cancers that were not driven by the hormone gene pathways. And breast cancer was the one that first led the way at identifying that there was variability. Now, you never have to look under a microscope ever again, and you never have to have looked under a microscope to look at these 8 different breast cancers that are depicted on the screen to see that they don't all look the same. They look different. And they look different because, in fact, they are different. But they're all breast cancers, all came from the breast and all cause the problems. Our pathologists can tell us if they're invasive or in situ. They can tell us whether the malignant cells remember they're supposed to form glands, so they're trying to form these circles, that would be the ducts, versus where you see poorly differentiated, the malignant tissue doesn't even remember what it's supposed to be. And they are more aggressive than the well-differentiated. The nuclei look certain ways, and they can describe those, the margins, the cells that are infiltrating. But what we really are hoping is that with the new explosion in biotechnology and molecular biology and our ability to look at the DNA, that we can use this new molecular information to better identify the subgroups. So we need a paradigm shift, and that paradigm shift is this. Number one, cancer is not a single disease, so you can't just say the patient has cancer and there's one treatment or a silver bullet. But and this died a long time ago, this concept that cancer was a single disease. What died a little harder was this, that cancer within a specific organ is not a single disease, but maybe a spectrum of diseases or several subtypes. And then we need to adjust our therapy based on that information. Now, what we've been doing at UCLA and some of the things that we're trying to take to the community in terms of moving from the lab to clinical practice and translating stuff is to do experiments to tell us what identifies this heterogeneity of the disease. What is the molecular heterogeneity? So we have a panel of 51 breast cancer cell lines, each one from a different patient, and it's 51, so it's a large enough number that we think we're getting some representation of the real spectrum of the disease. And we were lucky enough in 1987 to be involved in some work where we identified a molecular alteration, one of these mistakes that the spell checkers don't pick up, in the DNA, in a particular gene called the HER2 gene, HER2. Now that gene has nothing to do with gender. HER doesn't have to do with gender. It has to do with something called the human epidermal growth factor receptor number 2. Scientists are very clever. There was one discovered before it that was called number 1. There were 2 discovered afterward. They were called 3 and 4. So much for advanced technology and nomenclature. But these genes code for a protein that is a growth factor receptor. It's something that sits on the surface of the cell and receives this. Now, I have enough gray hair that I can show a picture like this. Some of you remember something called a television antenna that used to sit on the roof of your house. That's passé now. Everything's done through cable and through satellites. But that television antenna used to receive television signals. Radio antennas would do the same thing and transmit the signal down to your television set. Well, think of the HER2 gene as the antenna. It receives signals from the outside telling the cell to grow. And it's this completely regulated, carefully orchestrated growth that's supposed to happen every month, supposed to happen with pregnancies. This gene plays a critical role in it, as do the endocrine genes and hormone receptors. But if it gets broken, which is what you see in this picture that I've just shown you, you have a problem. Now, again, you don't have to be looking at sophisticated blot technology, which is what this is, to really see a difference that I'm going to point out to you at the very top of the figure. At the very top of the figure, there are 5 different tumors from 5 different women. We put the exact same amount of DNA in each lane, and then we've probed it for the HER2 gene. And what you can see is going from the right to the left is the signal increases in intensity. That is indicative of one of these mistakes. In those breast cells, The Xerox machinery that copies the blueprint made too many copies of the region of the chromosome that contains this receptor, and you have too many growth factor receptors on the surface of the cell. Does that make